So I want to show you how to find the information about which mutation your team will want to select. Remember that you are trying to target a location on the CFTR protein so that it can function in its normal job of moving chloride ions out of cells and preventing buildup of sticky mucus in lots of different places, including the lungs as well as any other duct in your body, because this protein, unlike many others, is expressed in every single cell in your body. So you're going to see the bad effects of a mutated protein in lots of different places, which helps explain why there's so many different kinds of symptoms of cystic fibrosis, not just lung clogging. So I've gone to this site called CFTR2.org. That's what I typed into my URL bar here. It took me to this site that's, you can see that there are several different reputable sources, arguably, I guess, that support this site. And I'm going to show you how to use it. So when you go to click on Start Here to search, and then search the mutation database, you have to, what you'll actually see is I've already done this once today. You'll see 4 boxes to check, agreeing that this is for educational purposes, that this is not for diagnostic purposes, that kind of thing, and agree to those things because we're using this for educational purposes. And then you'll get to this screen, and this screen you could type in a mutation notation if you know it, but you may not, and maybe the best way to go is to actually click just on Show All. And here I've got lots and lots of different mutations that cause cystic fibrosis, and all these mutations appear on CFTR. Some of these are better targets than others, and now it's your job to figure that out. So let me explain to you the notation. So this first one is one that you're not going to be able to do. It's a deletion of the 508th amino acid. So F stands for phenylalanine, which is the 508th amino acid in this sequence, or the 508th amino acid, which happens to be phenylalanine, is deleted. Now this is in the protein, so it's actually the codon in the DNA that was deleted, and therefore this amino acid is missing, and so that changes the whole structure of the protein. But this is actually considered a type 1 or class 1 mutation, and you're not going to want to model a class 1 mutation, because what happens is the protein doesn't fold correctly, and it gets targeted to be broken down and recycled for parts, and the cell, having the exact same DNA it did the first time making the mutated or the altered protein, will make another altered protein. So it's this cycle where basically the CFTR protein never makes it to the membrane to do its job. That's a class 1. You don't want to do that. But over here is another notation of it, and what it's telling you is that the 1521st through the 1523rd nitrogen base, which happens to be CTT in the DNA sequence, was deleted. And that's how you read one of these notations. And we're not looking at the DNA sequence, and I can provide that to you if you like. Just ask me, and I'll provide that resource to you. Otherwise, we can look at the amino acid sequence and determine how to model that. So if I were to show you where the 508th phenylalanine is, actually you could do that now once you have the JMOL, the PDB file. You could find the 508th amino acid and turn on its wireframe and space fill it to see it on an alpha backbone. You might want to look at some of these others. Something important to look at is not duplicating ones that already have treatments for them, so you might need to figure that out, what treatments are out there and for which mutations. But here's another source of information for you, cftrscience.com. It's a .com. This just gives you a little bit of background on different types of CFTR mutations. This is the normal functioning CFTR. The DNA is transcribed in RNA, then translated at the rough ER, because this is a protein that's going to be leaving the body of the cell, goes to the Golgi complex, might get modified, and then sent to the cell membrane, the apical surface of one of these cells, and it's letting sodium ions out. It is ATP triggered, and that's important because that's an important binding site. I'll provide you a resource to help you find that in your protein. But if it is misfolded, it never finds its way over here. It just kind of cycles back and gets reused for parts. If it's misformed, a proteasome out here might chop it up into pieces and recycle it. And so when you're looking at different classes, you're really going to look for anything but class 1. Class 2 does get to the surface in lower quantities, but there are some problems here. Class 3 is a little bit better. Class 4 and 5 and 6 have other issues to deal with. And so going to this website for mutation classification would definitely be worth your while, and to determine which of these mutations, you're going to have to use another source here, which of these mutations are the right class, and then you're going to have to model in JMOL to make sure that that protein is on the outside of the protein so that the drug can target it and interact with it successfully. Okay? You also want to look at the prevalence of that mutation. How many people would it actually help to develop a drug for that treatment? Okay? Then I'm going to stop there.